So let us draw up a table of oxidative phosphorylation and the ATP yield. So let's look at all the processes beforehand which provided the electron carriers, beginning with glycolysis. So glycolysis, if you remember, produced two NADHs. So now, if NADH, the electron carrier, is in complex 1 will pump out a total of 8 hydrogen ions from the matrix into the intermembrane space, because for one NADH it's 4, remember. And then for complex 2 nothing is pumped out. Complex 3, 4 hydrogen ions gets pumped out into the intermembrane space for each NADH. So because it's 2, it gives us 8 hydrogen ions. And then for complex 4, 2 hydrogen ions are pumped each for each NADH, so 4 hydrogen ions. So the total hydrogen ions pumped by 2 NADH molecules from all the complexes equates to 20 hydrogen ions. So now we have 20 hydrogen ions in the intermembrane space from 2 NADH H's, the electron carriers. And so if we put this through the ATP synthase, where 4 hydrogen ions will give us 1 ATP, this would mean that 20 hydrogen ions will produce 5 ATP. Similarly, with the preparatory step, which gave us 2 NADHs, this means that it's exactly the same. Complex 1 will pump out 8 hydrogen ions into membrane space. Complex 3, 8 hydrogen ions. Complex 4, 4 hydrogen ions, giving a total again of 20 hydrogen ions. And so the total amount of ATP produced from 20 hydrogen ions is 5 ATP, because 20 divided by 4 is 5. Looking at the Krebs cycle, if you remember, produced 6 NADHs. And so if 6 NADHs goes through complex 1, this means that it would be 6 times 4, because for each NADH, 4 hydrogen ions are being pumped from the matrix into the intermembrane space, which means that 6 times 4 is 24 hydrogen ions, which means complex 3 also 4 hydrogen ions are pumped for each NADH, so 24 hydrogen ions in total. And complex 4, finally, 2 hydrogen ions for each NADH, so 6 times 2 is 12 hydrogen ions, giving a total of 60 hydrogen ions, which are pumped from the matrix into the intermembrane space. And so how much ATP would this create? Just 60 divided by 4, because 4 hydrogen ions makes 1 ATP, giving us 15 ATPs in total. The Krebs cycle, however, also produces 2 FADH2s, right? So what about these? Because we learned about NADH only in this diagram. We didn't learn about FADH. So where does FADH come into the picture? Well, it doesn't start at complex 1, surprisingly. FADH passes the electrons from complex 2, which is succinate dehydrogenase. So now I will rub out all these hydrogen ions which were pumped out by 1 NADH. And so now we will not look at NADH, NADH's electrons moving through the electron transport chains, but we will look at FADH and how its electrons moves and how many ATP it will produce. So where does FADH2 come from? If you remember from the Krebs cycle, the conversion from succinate to fumarate requires the enzyme succinate dehydrogenase, which is our complex 2. And so through this conversion, FAD, which is part of complex 2, will be reduced to FADH2. And FADH2 is what provides the electrons to be transported through the electron transport chain. Now let's just look at the actual complex 2. Now complex 2 actually is comprised of 4 main parts, subunits, A, B, C, and D. We don't really need to know much about these, but essentially in the center somewhere we have another series of iron centers, which the electrons from NADH2 will go through these iron centers and finally to ubiquinin. So the 2 electrons will go to ubiquinin, and ubiquinin will turn to ubiquinol. Now ubiquinol will then travel to complex 3, cytochrome C oxyreductase, where it will pass on the electrons to cytochrome C. During this process, complex 3 will also pump 400 ions from the matrix into the intermembrane space. Cytochrome C with the electrons will then travel to complex 4, cytochrome oxidase, where at complex 4, it will cause an additional 2 hydrogen ions to be pumped out from the matrix into the intermembrane space. So, as you can see, the transporting of electrons from FADH and NADH are very similar. However, FADH does not go through complex 1. NADH pumps 10 hydrogen ions in total through the electron transport chain. However, FADH only pumped 6 hydrogen ions from the matrix into the intermembrane space. Therefore, if 6 hydrogen ions were to go through the ATP synthase pump, it will only produce 1.5 ATP because 4 hydrogen ions makes 1 ATP. And so as you can see, FADH will only produce 1 less ATP than NADH would because FADH does not pass through complex 1. And so if we go back to our previous table of our ATP yield, we look at the 2 FADHs and look at how many hydrogen ions it will pump. At complex 1, it does not go through it, so it doesn't pump any hydrogen ions. Complex 2, it doesn't pump any hydrogen ions. Complex 3, it will pump out 4 hydrogen ions each, giving a total of 8 hydrogen ions. And so also complex 4, 1 FADH will pump out 2 hydrogen ions, so 2 FADHs will pump out a total of 4 hydrogen ions. So the total hydrogen ions pumped from the matrix into the intermembrane space by 2 FADHs is 12 hydrogen ions. So how much ATP can 12 hydrogen ions produce? Well, we go 12 divided by 4, which will give us 3 ATP produced by 2 FADHs. Now, to make things a bit more confusing, in glycolysis, because the 2 NADHs are coming from the cytoplasm, not from inside the matrix, and so the 2 NADHs from glycolysis have to have some form of mechanism to move from the cytoplasm inside the matrix, right? One of its way is through shuttles. There are 2 types of shuttles. One of them will cause the electron transport chain to not pump as much hydrogen ions from the matrix into the intermembrane space. And because this particular shuttle, the 2 NADHs from glycolysis, will miss going through complex 1. And so therefore, the amount of hydrogen ions pumped from complex 1 can vary from 0, 4 to 8 hydrogen ions. So the total amount of hydrogen ions pumped will technically be either between 12 and 20 hydrogen ions. So the amount of ATP produced can vary from 3 to 5 ATP for 2 NADHs from glycolysis. I hope that makes sense. And so why is this again? It is because these 2 NADHs are coming from the cytoplasm and need some form of mechanism to go inside the mitochondria. One of its mechanism will cause it to miss going through complex 1 of the electron transport chain. And so this will cause not as much hydrogen ions being pumped. So let's look at this particular shuttle. And the shuttle is known as the glycerol 3-phosphate shuttle. It is called glycerol 3-phosphate shuttle because it involves 2 enzymes known as glycerol 3-phosphate dehydrogenase. And the 2 enzymes are actually different in that one of them is situated in the mitochondrial inner membrane, such as here, and the other one is in the cytosol, in the cytoplasm. And so what happens is NADH will come from glycolysis. We're only looking at one NADH. NADH is brought into the electron transport chain when it reduces dihydroxyacetone phosphate into glycerol 3-phosphate with the enzyme cytosolic glycerol 3-phosphate dehydrogenase. And so now we have glycerol 3-phosphate with the 2 electrons, you can say. Now glycerol 3-phosphate will then be oxidized by the same enzyme, except in the mitochondria, glycerol 3-phosphate dehydrogenase, the mitochondrial one. And during this process, FAD is reduced to FADH2. And FADH2 is what provides the electrons to ubiquinin, converting it to ubiquinol. Ubiquinol will then move the electrons to complex 3, while complex 3 will then pump out 4 hydrogen ions per NADH, now FADH2. And then the electrons will move to cytochrome C of complex 3. Cytochrome C will then move to complex 4, where it will cause complex 4 to pump out 2 hydrogen ions per NADH, now FADH. And so as you can see, through this glycerol 3-phosphate shuttle mechanism, we are passing complex 1 and complex 2. So we're passing an additional of 4 hydrogen ions pumped by complex 1. And another amazing thing is that we begun with NADH from glycolysis, but we end up with FADH2, which will provide the electrons to ubiquinin. And as we know, FADH will actually produce one less ATP than NADH would. And this all makes sense when you think about it. So now, if we go back to the graph, we can see that the total ATP from oxidative phosphorylation produced from essentially one glucose molecule, we can say, it equates to 26 to 28 ATPs, depending, remember, if we use the glycerol 3-phosphate shuttle. I hope this all made sense to you, and I hope you enjoyed this video. Remember, I might have made some mistakes, and another thing is that this electron transport chain, oxidative phosphorylation, is not actually fully understood yet, so it might sound all weird. Thank you for watching. Please like, subscribe, and